Hey guys, this is Sasank from Biosimplified. This video provides a structured overview of immunology to help you understand the fundamental principles of how the immune system works. So, the goal of this video is threefold: to introduce the core concepts of immunology clearly, to serve as a big picture reference that you can revisit after learning the finer details, allowing you to integrate complex information into a simple framework. So, broadly, the immune system is divided into two parts: the innate immune system and the specific or adaptive immune system. The innate immune system consists of components that are already present at the site or location of infection, and it responds immediately but in a generalized manner. On the other hand, the adaptive immune system involves specialized B and T lymphocytes. These cells reside primarily in lymphoid tissues away from the site of infection. Once activated, they mount a highly targeted response against a particular pathogen. Because this system requires recognition and activation, it takes longer to respond, especially during a first encounter with a pathogen. In this video, we will examine both these systems and also explore the complement system, which supports and enhances the actions of both innate and adaptive immunity. So, what happens when a pathogen enters the body? Before infection can occur inside our body, a pathogen has to breach several protective barriers. They are physical barriers and chemical barriers. First one is our skin, the primary protective barrier which most pathogens can never pass. Stratum corneum is the outermost layer of the epidermis, which acts as a brick and mortar shield that retains moisture and locks out external pathogens, irritants, and pollutants. Composed of hardened skin cells referred as bricks and lipids like ceramides, cholesterol, and fatty acids as mortar. If the skin is broken, example, a cut is there, pathogens may gain entry. Other physical barriers include respiratory mucosa, gastrointestinal lining, urinary tract mucosa. After physical barriers, we have our chemical barriers. The stomach, which has the gastric acid, has a very low pH, denaturing proteins and destroying most pathogen ingested with food or mucus. The lysozyme, which is found in our tears, saliva, and mucus, breaks down the cell walls of bacteria. In our reproductive pathways, like vagina, a very low pH environment created by lactic acid restricts the microbial growth. These mechanisms destroy or inhibit pathogens before they can establish infection. So they are coined into physical and chemical barriers. Moving on to the next. So now you must be wondering what would happen once barriers are breached. Three immediate events occur as initial immune response. The macrophages detect the pathogen and activate innate immune response. The dendritic cells pick up antigens of pathogens and travel through blood and lymph, locating and activating relevant B and T cells. The pathogen directly activates the complement system via the lectin and alternative pathways. So first, we'll see how the innate immune system provides immediate response to infection from any pathogen. When innate immune system comes across any pathogen, macrophages identify pathogens through structures known as pathogen associated molecular patterns (PAMPs). These are molecular signatures present specifically on microbes but absent from the human cells. Macrophages detect these PAMs present on the surface of the pathogens using special receptors present on its own surface, which are known as toll-like receptors or TLRs. So, what happens once recognized? Once recognized, the macrophages and other phagocytes like neutrophils perform a process known as phagocytosis. So, phagocytosis is a specialized form of endocytosis where immune cells, which are also known as phagocytes, like macrophages and neutrophils, engulf and destroy large particles such as bacteria, debris, or apoptotic cells by surrounding them with pseudopodia, creating a phagosome for degradation via lysosomal fusion. It is a critical receptor mediated defense mechanism in the innate immune system, essential for tissue homeostasis and initiating immune responses. So, the phagocytosis is done in majorly in four steps. The first one is engulfment, then phagosome formation, third is digestion, and then finally elimination. So, we'll discuss about each one of this process one by one. The first step is recognition or detection, in which the phagocytes recognize targets via surface receptors known as TLRs or toll-like receptors, which bind to pathogen-associated molecular patterns PAMPs. The cell membrane forms pseudopodia, which are temporary cytoplasmic arms around the target particle supported by the cytoskeleton, and the pathogen is engulfed into the cell. The pseudopodia fuse, trapping the particle inside a membrane-bound vesicle called a phagosome. The phagosome fuses with a lysosome, forming a phagolysosome. Hydrolytic enzymes, reactive oxygen species, and low pH kill and digest the pathogen inside the phagocyte, and the pathogen is degraded into harmless waste. If macrophages can control the infection alone, the process ends here. But if the pathogen load is too large, additional immune support is required. In this case, macrophages release cytokines, which are small signaling proteins that function as chemical messengers. These molecules coordinate communication between immune cells and orchestrate the inflammatory response. Cytokines act on nearby blood vessels, immune cells, and surrounding tissues to initiate inflammation and recruit additional defensive cells to the site of infection. Cytokines and chemokines create a chemical gradient that directs immune cells towards the infected tissue, a process known as chemotaxis. The main recruited cells include additional macrophages to increase phagocytic capacity, monocytes, circulating precursors in the bloodstream that migrate into tissues and differentiate into macrophages. Then there are neutrophils, rapid response phagocytes that arrive early and aggressively destroy pathogens using enzymes, reactive oxygen species, and antimicrobial granules. This influx of immune cells significantly amplifies the local immune response. Inflammation is a coordinated vascular and cellular response aimed at containing infection, eliminating invading pathogens, and initiating tissue repair. It is characterized by vasodilation mediated by prostaglandins, nitric oxide, and histamine, which increases blood flow to the affected area, producing redness and heat. Tissue resident mast cells rapidly degranulate, releasing histamine, heparin, and more cytokines. Simultaneously, increased vascular permeability occurs as endothelial cells contract and create gaps, allowing plasma proteins, antibodies, and immune cells to exit the bloodstream and enter tissues, resulting in swelling. The local activation of the coagulation cascade generates fibrin clots that restrict pathogen dissemination and form a temporary physical barrier around the infection. In the parallel, activation of the kinin system produces bradykinin, which increases vascular permeability, promotes vasodilation, and induces pain, the latter serving a protective function by limiting movement and preventing further damage. Inflammation itself actually stimulates macrophages and neutrophils to secrete more cytokines, notably chemicals called interleukins. This is known as acute phase response, and it leads to a more systemic inflammatory response. This involves sending cytokines, specifically interleukin 1, to the brain to tell it to produce a fever that leads to high temperatures that are poorly tolerated by many pathogens and cause reduced appetite and lethargy, so that the person conserves more energy that can be used to fight infection. It also involves sending interleukin 6 to the liver to produce acute phase protein that acts to something called opsonins. To keep this word in mind, we try to keep this word in mind because we'll talk about opsonins in a minute. Additionally, interleukin 8 is released that recruits and activates more neutrophils. And then at last the interleukin 2 as well as 12 activate natural killer cells, and tumor necrosis factor alpha is released that does all of these effects by itself. As I just mentioned, I wanted to take a quick look at opsonins as these are very important in the immune system. Opsonins are complex extracellular proteins that bind to pathogen and enhances phagocytosis by helping neutrophils and macrophages. You can think of them a bit like a fork that is pierces the pathogen so that macrophage can attach to the other end of the fork and then use it to eat the pathogen. The word itself means to prepare for eating in Greek. Apart from marking pathogens, opsons also mark dead or dying cells as well as infected and tumor cells. Antibodies are an excellent example of opsonins. Another example which you might also have heard of is the C-reactive protein or CRP, which is produced by the liver in response to interleukin 6, and we actually measure the level of CRP in patients to assess how much inflammation, if present, there is in the body, and it can be a really good indicator of severity of infection that the patient is suffering from. And hence that summarizes the innate immune system, a generalized system that recognizes and responds to an invading pathogen by causing an inflammatory response and recruiting cells that destroys the pathogen by phagocytosis. Next, we will look briefly at the complement system. The complement system works alongside the innate and the specific immune system to help them destroy pathogens. This involves a series of complement proteins labeled C1 to C9. Once a complement system is triggered, the protein starts to activate each other in something called the complement cascade, and various products of the complement cascade have important functions such as opsonization to increase inflammation and direct pathogen destruction. The complement system is triggered in one of three pathways. The lectin and the alternative pathway are activated directly by pathogens, and the classical pathway is activated by antigen antibody complexes that arise from the specific immune system. And that brings us nicely on to looking at the function of the specific immune system, which involves two characters, the T and B cells, and these are both lymphocytes. Now, this was the first part of this video on the overview of immunology, in which majorly the innate immune system was explored in detail. In the next video, we will be learning about specific or adaptive immune system in detail. So, stay tuned for the next video. Thank you.